Thank you for all the organizers for this invitation. I'm very proud to be here. And so I will show you today some work that we have been exploring together with Hartmut Leuven and some other collaborations, as you see. And so basically, I will show you vertical transport above active carpets. So I will start first with the motivation. So we will consider flagellated organisms moving in an aquatic system, okay? So this is different from the last talks in the sense that they are neglecting hydrodynamic interactions between active matter. And in this work, we will consider an aquatic environment. So our microswimmers are moving in a fluid, and they are exerting, actually, let me see if I can use the pointer. Okay, and they are exerting a flow field in the media. So for us, an flagellated organism is a microswimmer, such as a bacteria, which is a micro-sized particle, which is moving persistently in some direction and performs random motion while they are moving, okay? So for us, microswimmers are, or we know that microswimmers are attracted to surfaces. So when they are moving close to boundaries, they are attracted to them, and they perform long time residency around these surfaces. This is due mainly to four, let's say, reasons. The first one is that when microswimmers are close to solid boundaries, they perform circle motion or chiral motion in the boundary. The second is the chemotaxis, that due to the chemotaxis, they form patches on the surfaces. Also, due to hydrodynamic attractions to surfaces, they accumulate in the non-slip boundaries or in the solid surface. And finally, due to the formation of biofilms, so we know that microorganisms such as bacteria in the early stages of the biofilm formation, they are still active and they can move, and they are actually moving above some solid boundary, let's say. So we will consider that, okay, microswimmers spend long times above surfaces, they like to swim close to them, and they are also forming like interesting architecture above the surfaces. That's why we defined active carpets. So this is a work together with Professor Hartmut Leuven and also Arnold Matthijssen from the University of Pennsylvania. So what we do in our active carpets is that we consider a flat boundary. This flat boundary can be a solid one or maybe a fluid-fluid interface. And we consider microorganisms moving above the surface, okay? So in the case of bacteria, we can imagine that is bacteria in the early stages forming a biofilm. So they are moving in this solid surface and exploring in 2D, let's say, the surface. We can consider attached cilia to a surface too, or these kind of extensors called vorticella, which is they are attached to a surface and they are performing vertical motion. And finally, we can think an active carpet as an algal bloom or a floating film of microalgae, okay? So let's be more specific. So for us, the active carpet, it's we can consider like in terms of simulations as a flat boundary with periodic boundary conditions. So our microswimmers are allowed to move only in 2D. So they are moving parallel to this surface, and they are exerting flow fields. The flow fields are only steering the fluid above the active carpets because we are not considering hydrodynamic interactions between the microswimmers. So the microswimmers are only moving and forming these nice architectures, but they are not interacting between microswimmers, although they are steering the fluid, and we can measure how larger is these fluctuations or this steering when we put a probe particle at different heights, okay? So our proof particles or tracer particles, we will put them at different heights above the active carpet, and we want to see how they move. So that is the main question, how they move, how they diffuse above these kind of systems. So we will start with first with an active carpet with self-organization. Then we will consider an active carpet in a random configuration. And then we will consider an active carpet in a fluid interface. And finally, we are wondering if active carpets are or can start aggregation process in the microscale. So this is the final thing that we are exploring. So first thing, so we now consider our flat surface as a non-slip boundary, okay? So now our microswimmers are moving in 2D above a non-slip boundary. We have to solve, in order to find the flow field for the bacteria, we have to solve the Stokes equations with the proper boundary conditions, and then we can measure, for instance, the average flow field of the collection of microswimmers at different heights from the active carpet. In this stage, we will consider two general cases for our carpets. We will consider carpets or active carpets that has density gradients, in the sense that they are not homogeneously moving in the surface. So microswimmers are doing self-organized movements. And also we will consider the case when we have orientational gradients between microswimmers. Okay? So if we consider that, we found that the average velocity above the active carpet is different from 0, and we found a very nice results. So if we consider the easiest case, so when we have a cluster formation of microswimmers. So now we are considered, let's say, chemotaxis. So all the microswimmers want to go to one hot spot, and they are accumulating there, and they are leaving a density gradient, let's say. So in the gray area, we can say there are no bacteria moving, but in the green vortex or in the green cluster, we have bacteria moving there. Okay, so if we measure the average flow field above this active carpet, we found that above the microswimmers, we found a very large drift towards the active carpet, and also we found a region of repulsive flow. So this attractive and repulsive flow depends, of course, on the size of the cluster formation, and we have that in the case of a very large clusters, the drift will disappear. And the drift, as you can see here, depends on the size of the cluster and also on the height at which you are measuring the flow field. So this is basically what I'm saying. So we consider that the bacteria are the black dots, and they start to make a cluster formation due to chemotaxis, for instance. And you can see that all the tracer particles in colors, they start like randomly distributed above the active carpet, and then when swimmers are accumulating here, you can see that the drift appears. So for us, we can consider that this is a way of bacteria, again, so this is the way of bacteria can attract nutrients, for instance, for them, okay? And we found that this drift or attractive flow, it doesn't only appears in the cluster formation, it also can appear in bacterial turbulence. where you can see areas where we have like the repulsive flow in red and blue areas where we have this attractive flow towards the bacteria. In the case of bacterial turbulence, this attractive and repulsive flow are due to orientational gradients. And also, we can see the orientational gradients here. In the case of vortex formation, so we have, again, like bacteria doing this swirling motion, like in this movie, and we can see that when bacteria is moving, like in this vortex motion, they form an attractive flow to, above the area where bacteria is located. Okay, so we now know that that these attractive flows has been measured recently in other configurations. For instance, for cilia, they measured that the cilia, a carpet made of cilia, can pump flow above the carpet. And this pumping is also attractive in the area where cilia is moving. So it's very nice results. And also recently, it's possible to form like very nice bacterial organizations that we think it might be interesting to test if they can measure the flow above these configurations, like the swirl motion or display configuration, to see if the theory that we develop is right with the experiments. Okay, so next step, we will still consider a flat surface with our microorganism above the in the active carpet, and we are still in the case of a solid boundary. So the black carpet is still a solid boundary, and now we will study a different setup. So now we will consider that our bacteria are homogeneously distributed above the solid boundary. So we don't have an architecture now. We have just a random motion, random orientations, and now we will consider 4 different kind of microswimmers. So this is I didn't put the paper here. I don't know why. Yes, but here we study different flagellated organisms. So we consider cilium, bacteria, or vorticella, or molecular motors above the active carpet, and then we did something similar. So we consider them only moving in 2D, and in this case, our motion is what we call fast dynamics, in the sense that in our simulations, we will consider that at each time step, a new carpet is set up. So we consider a randomly orientated microswimmers in an homogeneous way, also in orientations and positions at each time step. So we are not allowing bacteria to move like a AVP, right? So we are like taking photos of the bacteria at different configurations in order to be or to study the diffusive movement of bacteria. So we are considering that the bacteria is like diffusing in the active carpet, and we want to see how is the transport above. So first, what we realize is that as we expect, the mean flow is 0, because all the bacteria is normally distributed, and the flow field then, if you measure at different heights, you will find a mean flow equal to 0 for the 4 different carpets. Nevertheless, if we measure the variance or the hydrodynamic fluctuations, we found that they are different from 0, and actually in almost all the cases, the vertical fluctuations are larger than the horizontal fluctuations. So in this step, at this step, I will remember you the configuration. So we have the active carpet with the microswimmers moving in the active carpet. They are not forming any kind of cell of organization. They are just diffusing in the system, and we are measuring the flow field above the active carpet. So we found that the mean velocity flow is equal to 0, but nevertheless, the fluctuations are not. So we observe that the vertical fluctuations are more important than the horizontal ones. And for us, that was a very nice result in the sense that we found that the active carpet can resuspend tracer particles when they are like falling into the active carpet. And for us, this is an important result for transport at the micro scale. We found that the particle distribution above an active carpet in this kind of configuration is not non-Boltzmannian. And yeah, so it's, for us, it's like a cleaning process. So in the first case, as you remember, we can have these attractive flow fields that allows microorganisms to eat or to reach nutrients. But in this case, when they are like just randomly moving, we are resuspending particles. So we are transporting particles above the active carpet. Okay, so what we are doing now. So now we are considering what happens when we have a floating film of microorganisms. So what happens when we don't have now a solid boundary, but we have a fluid interface, okay? And fluid interfaces are important in marine environments or yeah, where you can have like density gradients or viscosity gradients, okay? And we are wondering, okay, what happens when microalgae accumulates at these interfaces? Is it possible that they start aggregation process or not? How is the pair diffusivity now? How they can merge or they can steer the fluid in such a way that they can make trigger aggregation process? Okay, so what we are considering now is an active carpet above a fluid-fluid interfaces. In our case, the fluid interfaces is a viscosity interface. So we consider one fluid here with viscosity mu1 and a different fluid here with viscosity mu2. And we will study this parameter lambda, so the ratio between the two viscosities, and how is again, the vertical transport above the active carpet. So we are interested in this area. What happened with particles in this area? How they are moving when this microalgae is moving or forming this floating film. So we again solved the Stokes equations. We found the flow field. And we know that why we are interested in these viscosity gradients or viscosity interface, because it has been studied recently that microorganisms such as chlamydomonas or microalgae, they accumulate at viscosity gradients, let's say. Okay, so this is the configuration, and what we found is this. So in this case, as you remember, this is the variance or the velocity fluid fluctuations, the velocity fluctuations. And if we consider that the active carpet is almost like here, let's say at this distance from 0, we again observe that the vertical fluctuations are larger than the horizontal fluctuations, but at some point when we are going further from the active carpet, we found that a crossing on the fluctuations. So the fluctuations at some point and closer to the free surface are larger in the horizontal direction than in the vertical direction. So this is something new, and this might be important for aggregation process, because now we found that the parallel fluctuations are larger than the vertical ones. So this might, now the steering of the fluid is larger in this direction than in this vertical direction. And we found that this cross in the fluctuations depends on the lambda parameter, so on the viscosity ratio, and also on the fluid, the film height, so H, this parameter H. Okay, so when we can consider like a thin film when the free surface is very close to the active carpet, or like a normal film when the free surface is farther from the active carpet. Okay, so yeah, so it's a space parameter much more complicated, but the important thing is that that the dependence on the viscosity ratio, we can move this critical or this point where the fluctuations are crossing. We can move it closer to the active carpet or farther from the active carpet. And the last thing that we try is, okay, actually we put some tracer particles in this way, so in a 2D carpet too, and we allow the colloidal particles or the tracer particles to move only in 2D, and we studied the aggregation process in this case. So again, our particles are only allowed to move due to the hydrodynamic distortions, and what we did is to count the encounters. So we count the particle encounters, okay? So we can consider two different tracer particles at some distance d0 between them and at some height zeta 0, and we measure two things for these two proof particles. We measure the velocity correlations and the pair diffusivity, okay? So in the case of the velocity correlations, we found, as you can see here, that the velocity correlation depends on the height at which these two particles are located above the active carpet. And yes, so we found a similar result as for bacterial turbulence. But this is the important one. This is what I want that you understand. So in the case when we put the two particles and we measure the pair diffusivity, we found that the pair diffusivity not only depends on the distance between the two particles, it also depends on the height. So we found that pair diffusivity is larger when you are closer to the active carpet. So when you put two particles very close to the active carpet, they diffuse much more than when they are farther from the active carpet. And also, if we count the encounters between the particles, so now if we put the two particles and we measure the time that it takes for these two particles to collide, we found that you can have more collisions when you are close to the active carpet, and also when you have larger particles. So for us, R0 is the size of the tracer particle, and again, well, and D0 and zeta 0 is the height. So this is the message. We can have aggregation process. We are not sure still if this is very important, because this is just the first results. So now we have to compare with other systems to say something about this aggregation process. But what we know now is that aggregation process appears above active carpets. That is anisotropic in the sense that it depends in which direction are you looking at the aggregation process. It's also larger close to the active carpet, and it's getting more difficult to aggregate particles as you go farther from the active carpet. And finally, that you can aggregate larger particles than smaller particles. So yes, that's it. And thank you very much for your attention.
Thank you very much, Francisca. I have a question myself. So when you showed the situation of a green area where the swimmers were, and the nutrients were entering or going towards it, you can think essentially that there is a cooperation process there, right? So when they are together, they are capable of attracting the nutrients.
Yes.
But at the same time, you can think that there should be some competition, right? So they should compete. There is a lot of people there. They are capable of attracting a lot of food, but at the same time, there is a lot of people, so there should be some competition. Have you thought along those lines at some point, or?
No, we haven't thought about to put like on the above of this process like a competition, let's say equations or something like that. But what we can think about it is that when you are in a crowded environments, it's better if you organize than if you are not organized, if you want to eat. In that sense, like, this is the message. So in these very crowded environments where you have like a lot of bacteria or people moving, if you are organized, like forming these cluster formations or moving in the same direction, you are allowed to attract nutrients toward your position or where you are. But if you are moving like randomly, as in the second case, you are allowed to clean the system above you. So you can resuspend particles above and move them. So this is a very nice, there is people from theoretical biology asking me questions also about that, like, oh, do you think that bacteria has a sense, like, is this ecological or not? And yeah, it's a very nice problem. Thank you.
And one last question, myself as well. So the values that you used in the simulation, are they experimentally consistent values? The ones that you used to see that there is indeed enough force to rapidly suck the nutrients towards the carpet?
You mean in terms of experiments, if it's possible to observe?
If the parameters that you have used are somewhat realistic?
Yes, I mean in the second case, when we have this transport process above the active carpet, we measure some like typical characteristical flow fields and particles and particle size and so on. And we realized that this active transport is much more important than sedimentation processes or Brownian motion. Yeah.
So even if the active carpet's here and there is gravity against the thing, you should still be able to...
in that case, we are in the other direction, so the active carpet is here and particles are following by gravity like here, like this.
But even if it were the other way around, it should be, the hydrodynamic attraction should be capable of...
Yeah, it should.
Okay, thank you very much.
Thank you.